Good morning, teachers and friends. Now, me and Matthew will present about genetic technology.
What is genetic technology or genetic engineering? Genetic engineering is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. Genetic engineering involves the direct manipulation of one or more genes, and most often a gene from another species is added up to an organism's genome to give it a desired phenotype. Genetic variation can be defined as a genetic makeup of an organism within a population change. Genes are inherited segments of DNA that contains codes or the production of proteins. Genes exist in alternate versions or alleles that determines distinct traits that can be passed on from parents to offspring.
Human genetic. Human genetic variation is the genetic difference and among population. There may be multiple variants of any given gene in the human population allele, a situation called polymorphisms. No two humans are genetically identical, even monozygotic twins who develop from one zygote have infrequent genetic difference due to mutation occurring during development and gene copy number variation. Difference between individual, even closely related individual, are the key to techniques such as genetic fingerprinting. As of 2017, there are a total 324 million known variants from sequenced human genomes. As of 2015, the typical difference between an individual genomes and the reference genomes was estimated at 20 million base pairs, or 0.6 of the total 3.2 billion base pairs.
Human genetic population that are more geographically and ancestrally remote tend to differ more. The differences between the population represent a small proportion of overall human genetic variation. Population also differ in the quantity of variation among their members. The greatest divergence between population is found in sub-Saharan Africa, consistent with the recent African origin of non-African populations. Populations also vary in the proportion and focus of integrase genes they receive by the organic admixture both inside and outside of Africa.
Animals, bird. Level of genetic variation among individuals may affect performance by reducing the ability of prey to detect and escape from predators. If lack of genetic variation, reducing flight ability directly or incredibly through reduced parasite resistance. We investigated vulnerability of common avian prey species to predation by the sparrowhawk, Accipiter nisus, and the goshawk.
Animals. Gentilis in relation to an index of genetic similarity among adults of potential prey species. We estimated the prey vulnerability index that reflects the abundance of prey relative to the expected abundance of according to local population density, and it is related to the index to pen sharing coefficients based on the analysis of mini satellites for adults in the local breeding populations. The prey vulnerability index was positively correlated with the pen sharing coefficient in both predators, even when controlling for potentially confounding variables. These findings indicate that the prey species with high pen sharing coefficients and hence low levels of genetic variation. They are more readily caught by the avian predator. Therefore, predation may constitute a major cause of low levels of genetic variation and extent of population of prey.
Example of genetic technology. Golden rice. Golden rice is a genetically engineered variety of rice, Orchia sativa. It has been modified by inserting a gene from maize and a gene from bacteria found in soil, which allow the plant to biosynthesis beta-carotene in the edible part of rice. Beta-carotene is naturally found in many fruits and vegetables, such as squash, papaya, and carrot, giving them their yellow color, and it can be synthesized by the human body to make vitamin A. Characteristically, this genetic modification make the rice gain a golden yellow color.
Golden rice was originally conceived by Professors Ingo Rodriguez and Peter Bayer, and it was their desire to donate golden rice as a gift to resource poor farmers in the developing countries. Early working during the 1980s on the golden rice was funded by the Rockefeller Foundation and the European Union. And in 2000s, the investor established a public-private partnership with agricultural technology company Syngenta to help further develop golden rice. And part of the relationship with the Syngenta was involved in gaining patent around golden rice, but they also carried out additional laboratory, greenhouse, and also the field research to help raise the beta-carotene levels. Golden Rice 2 was announced in 2005, which produces up to 23 times more beta-carotene than the original golden rice. At this yield, it is calculated that 144 gram, or around roughly 1 cup of golden rice, would have to be eaten to receive a recommended daily dose of vitamin A. The Golden Rice Project now operates under a humanitarian board within the International Rice Research Institute, or we call it IRRI, and they have been working together since 2006. Thank you.
Wes ah.
Wis.
Cik, tak lihat sih.
Ulang
Slide
slide
recordingnya.
closing awal tuh.
Apa?